we've got quite a lot of questions here. Some were sent in advance, and we've got some on the in the comments. So let's start off with the ones in the comments. Actually, before we do that, I thought your description of what happens in PSC in the bile ducts right at the beginning was excellent, and we should make that a little short video to share. It's really good.
I'm really pleased you liked it. Good. That was the idea. And as I said, I'm sure that for some of my colleagues will be 10 times better as we are explaining it properly.
It was very good. So first question, can you create artificial strictures in the lab so that drugs can be tested directly on them?
That's a very good idea. This is what we are trying to do now in the lab. This is our next line of research. So I'm very pleased to hear that we're thinking along the same thoughts. And well, maybe we can get you in the lab. This is a really cool idea. This is what we're trying to do. We're trying to use that's, I'm sure I'll put in the screen as well. Who's keeping a note as well? Clearly, this is being addressed straight away. No, that went at the top of her list.
That would be amazing, wouldn't it? If you could not only go in and replace parts, but actually, because almost when you were talking about the small ducts, it seems if they've got strictures, that's harder to get up into. But if you can actually work to help get rid of strictures, presumably there would be some way for the organoids to go up into the tiny little ones.
Exactly. So this is such a huge topic. We are not experts on collagen deposition strictures, which is the stuff that makes up the tissue that makes up the strictures. But I think one of the biggest problems is that we use models, mice, for example, because we have to see what happens in the body. Whatever you have in a dish, you never have the complexity of the body. Because in the body, you have blood flow, you have germs that are part of the body, not in the bile, of course, but in the gut, and there's a lot of circulation of breakdown products from this bowel, these gut germs. There's a lot of aspects that you cannot reproduce in the lab. But one way or the other, it is, so that's why we always do a lot of our work in mice. But mice have a unique regeneration potential. They are 10 times more resilient than humans. So what we see is that a lot of antifibrotic drugs will work brilliantly in mice, and when you take them through to humans, they just won't do that. So this is what we're trying to address, to generate a human model where we can see, can we now study fibrosis? And for that, exactly as you said, it's very important to have the whole tube, because the scar happens around the bile duct cells, not in the bile duct cell. Yeah. So if you model this whole duct and manage to make scars, then you can start testing what reduces progression of scars. I think it's a great suggestion.
I was wondering as well, you know, when you inject them in, and then we had the green cells and then the red ones, how do they know where to go? So why don't they just double up and, you know, make a double layer in some places? How do you know, how can you make them go in the right place?
Yes, so feel free to take any of those questions if you want. For this one though, because it's something that we worked on quite a long time, is that, you know, the body, it's the same way a blood clot knows where to form, where you've cut your hand. The minute that what's under the cells, the undercoating of the cells gets exposed, this is very sticky. So the cells that die leave off a surface that is so very sticky. And that very sticky surface attracts other cells. So if you were to sort of, how shall I say it? It's as if you have magnetic shoes and you've got a metal floor. If you start crowd surfing, probably your magnetic shoes will not hold you on the floor. But if there's a gap and you touch the ground, it will be very difficult to get off it afterwards. Do you see what I mean?
Yeah, so they're just attracted.
Yeah, it's so sticky. So what we call the stroma or the matrix, or the tube itself, put it this way, is extremely sticky for the cells. Whilst one cell to the other is not so sticky. So they can slide off each other, but when they fall in the gap with that sticky substance, which is the underlying, we call it stroma or basal membrane, but the underlying substance of the duct, they stick there and they cannot leave anymore. So in a sense, it would be as if you took a cobblestone where some of the cobbles were missing, and you took a track and you started emptying stones on the cobblestone, and then you swept everything off. The ones that fell in holes would just stay there. Everything else would be swept off. And the sweeping process is what bile does, because bile, don't forget, is flowing and washing everything off constantly.
Well, that must have been amazing when you discovered that that's how it would work, and you could just get it to fill in the gaps. I mean, that must have been amazing.
That was really exciting. So first time.
You must have had quite a lot of those like eureka moments during this whole development of these organoids.
I don't know. I'll let Teresa tell you about that. Teresa is the most young member of the lab, and therefore.
Well, the eureka moment is absolutely the best. That is.
Yeah.
But it's even better when you can share it.
It makes it worth coming in on Christmas Day, doesn't it, to feed them or look after them.
Absolutely. It pays back for all those late nights at work.
Is there a rejection issue with the injection of ducts grown in the lab? And this is a really good question, does the injection have to come to be grown from the recipient?
So, is there a rejection issue? We are testing that to answer it definitively, but theoretically at least, there is a rejection issue, absolutely. We don't have any suggestion that the bile duct is a privileged location from an immune perspective. There are areas like the eye or the brain where you can get more immune privileged cells where rejection is not such a big problem. But for the bile duct, we believe that rejection will be a problem. So do you have to grow your own cells? Yes, if you don't want to use immunosuppression, you absolutely would need to grow your own cells. And so the recipient cells will have to go back in the recipient, right? And you would be using self cells. So is that impossible to do? No, because there is always some cells which have escaped disease. Ultimately, there are areas which are often not completely affected by the disease. So in PSC, you could use a very small duct in a biopsy, or you could use even the gallbladder, albeit the gallbladder, we don't know whether it's not affected in PSC because we know that we get polyps, right? So it's debatable. But then definitely there are cells which haven't been terminally damaged, put it that way. And you can grow these up. And you can then inject them without immunosuppression, and that would be the preferred choice. And this is why I mentioned earlier on that this is not a treatment that is ideally done 5 days before transplantation, because it won't work, but also because you need some time to grow the cells. You need at least a few weeks, 8 weeks or something.
So they're not artificial, really, are they? We say artificial bile ducts sometimes, but they're not artificial, are they? They're grown.
No, exactly. The artificial bit of the bile ducts will be, if we use collagen ducts, for example, will be the collagen, the tubing, the thing that makes the tubes. The cells are human, and actually it will be your own cells if we're looking to do that without immunosuppression. Having said that, we know that we wouldn't necessarily give the immunosuppression we give for a liver transplant. As a matter of fact, it might be possible to just use what we call single agent immunosuppression. So if you've got autoimmune hepatitis, and some of our patients will know that who suffer from autoimmune hepatitis overlap syndrome, or some other of our patients who have had PSC even as children, would have this autoimmune sclerosing cholangiocytes, will have had immunosuppression. So it is possible to go on taking immunosuppression like azathioprine or mycophenolate mofetil or some other agent, and then take healthy cells on if it was super urgent to replace bile ducts, or if it was for some reason not possible to grow your own cells, which I think would still beat a liver transplantation, in the sense that you wouldn't have multiple agents because we use more than one agent for immunosuppression. And on the other hand, if something went wrong, it's much more easy to replace a small bile duct than a whole liver. If you see what I mean.
Is there an argument for somebody being diagnosed with early PSC for you going in to take some of their cells and saving them just in case?
I think that would be, yes, I think that's a great idea. Actually, Tom Carlson and Espen Melvin are already generating biobanks in Oslo, which is the leading center in Europe. The work we're doing there for PSC is amazing. So, and we've taken inspiration from them. And my understanding is that there are a lot of initiatives in the UK where we're starting to biobank these cells. And I think there's absolutely, I think it would be very, very good to be in a position to start banking these cells, so that if in the future you find out that now I could use some of my cells for repeated injections, now I'm reaching the time where I need cellular therapy, we can take out your vial of cells, pull them out, and start growing them and injecting them repeatedly. The same way people now use placenta, for example. If you have a baby, they ask you, do you want us to freeze your placenta? Reserve some of the stem cells for future use. So it could be another great idea, which I see Teresa has already noted down.
Yeah, there are so many good ideas that I keep keeping notes of everything.
We're gaining more than you are on this one. So.
Let's carry on, because there's some really, really good ones here. So this one is for the common bile duct. Why can't you just replace the tube with some artificial material? Like, why do you need the cholangiocytes?
Teresa has done the experiment here, so I can let her answer this one. So that, do you want to, Teresa? So that I don't steal the glory.
Yeah, of course. Yes. Thank you very much. The reason behind that is because, as Fotis mentioned, bile can be quite toxic and corrosive because it's used to break down food and fats. Therefore, when if you just put a material without cells that can sort of like create a protective layer, bile will eventually eat through this material. The same way in the stomach, the cells lining the stomach will produce some mucus to sort of protect the lining of the stomach. And if there is, for example, an ulcer, then the acids in the stomach will start eating through. The same thing would happen if you use just some material without any cell on. And the beauty of cholangiocytes is that if they're healthy, they are very resistant. And therefore, they can create this protective layer above any material where I can grow on. Therefore, we will need to add the cells to sort of prevent bile from seeping through.
And test that in the lab.
Yeah, we've done that with a very cool experiment in the lab.
So we put some bile on, so whether it gets through.
So the bile would cause even more damage, which we don't want.
Exactly.
That's a very good idea, right? I mean, it's very good as an idea. Common things being common, you need to start with the simplest experiment. That's what the MHRA advises all the time. So if you went to go to the MHRA and say, we've got a tube with cells, they would ask exactly this question. It's complicated. Why do you need the cells? So it was a very good question.
Yeah. You just thinking back to the mouse, that you showed a picture of the mouse, and you'd sewn in the little, I think, the collagen tube. Did you grow the organoids, the little bile duct cells, after you put the tube in, or had you already grown them for that mouse before you put the tube in? I.e., would you have a point? Would there be a time where you put a collagen tube in with no cells, but just wait for them to proliferate?
That's another good question. So we did put in a collagen tube without cells, and that mouse died, unfortunately. So it's in the supplementary data of that paper, and we've actually shown they got strictures immediately, because what happened is that bile ate through the tube, and then the mouse started producing a lot. Mice are very regenerative, so the mouse initially they didn't die straight away on the table when it got this collagen tube, but what happened is it produced a lot of fibrous tissue. It sealed up the hole in the bile duct, perforation as we call it, but then unfortunately it blocked its bile duct immediately. So after a while it became yellow and it wasn't happy. So we had to sacrifice the animal. So, and when we saw what was happening, it was exactly that. If the collagen tube didn't hold, and it was replaced by fibrous tissue, and all bile outflow was blocked. And to answer the first question, all these tubes were pre-coated with cholangiocytes, so we had already formed this protective layer so that they would be protected by bile when they go in.
How long would it take to get enough cells in a 10 centimeter piece of tube?
To grow them?
Yeah. So how long would it take for that to be ready to go in to the mouse or the human?
A few weeks, I think, but Teresa knows more than that. She does it day in and day out, right? So I will wait for her to confirm.
Well, that's the beauty of the system, because from a tiny sample, like a liver biopsy that really is tiny, you can barely see it sometimes. You can, in a matter of couple of weeks, you can have as many cells as you need for a small bioengineered bile duct, and that's what is fantastic, because in theory we could prepare it ad hoc. If someone plans an operation in a month.
This is from an existing line, right? But if we were to take someone's sample, it would take a few weeks to get to critical mass, right? And then we would see the tube. So yeah, but it's still a few weeks rather than several months. It wouldn't be 6 months to give you this idea.
Yeah. So this is possible to. It's quite good, isn't it, that it would be so quick? Like that's amazing, because it could be months and months. The next person says, are you looking for volunteers to trial things?
We will have to get approval for trials. We will absolutely be looking for volunteers to trial things. But not at this stage. We would need to make sure that before we try anything, we need to be dead certain that it will not give you any problems, right? Like when we get to clinical trial stage, we're looking for complications which are minor, not serious problems happening. So the first thing we need to do is, and we're doing now, is not only transplanting these things in animals, but following them for at least a year to make sure the cells do not change. We don't get cancer associated with these cells, because they keep growing and growing in the lab. If something grows so fast in the lab, you need to make sure it will stop growing in the body.
Yeah.
And it becomes difficult in larger animals. A mouse lives for 2 years. So if you put it in for, 3 months, it's an eighth of the mouse's life. If you put it in for 6 months, which is something we did after the paper, it's a quarter of the mouse's life. In human years, it's like keeping it in for 25 years and seeing that it hasn't had any complications. In a pig, it's a bit more difficult because you need a bit longer time. So we need to make absolutely certain that we hit these milestones, and then no huge risk before we start testing in trials for tiny complications. But we absolutely will be looking for volunteers, hopefully before not too long.
And actually as well, people can help already, can't they, by giving you their views on your research and helping you decide the research direction.
So there's gotten some amazing pointers already.
Yeah. You can always rely on that from PSC patients. They're brilliant.
This is very true, and I see that in clinic. It's an extremely well-educated group about the disease, and many times we get in this position where we hear about some latest development first from the patients, and we're very, very embarrassed having we haven't heard about that one yet.
We are. I think it's just something about the disease, isn't it? It makes us all want to know, how are we going to get better? How are we going to beat it? So we're all very alert to research. Okay, next question. Assuming human trials are successful in testing cholangiocyte infusions and replacing bigger bile ducts, if you were to guess, how long would it be before this type of treatment could become available to patients?
I would say that we need a couple of years to get MHRA approval. This is the most optimistic timeline, 2 to 3 years to get to the first trial. Most likely 5 years, which is a more sensible, relaxed timeline. And then the first trial would not be, you never do at first a trial that shows whether it's an efficient treatment. You first do a trial to show whether it's a safe treatment.
Yeah.
And what you tend to do is you put it in patients and hope that they do well and they tolerate it well, and then you put it head to head to the alternatives. You put it head to head, for example, to the normal biliary reconstruction surgery that we are doing now, which is called the Roux and Y photoenterostomy, right? If we saw something which really worries us in your bile duct, we want to take out just your bile duct, and it was in a position where we could take it out without giving you a new liver. If it was in a position where we had to give you a new liver, of course, the alternative is liver transplant, but then it is obvious that the artificial bile duct works better. But if we had to take it out without that, what we would do is we would take the bowel and bring it and suture it to where the right and left large ducts come together. And this is called the portoenterostomy, because this part where the ducts join is called the porta hepatis. So what we would need to do is show that it works better than a portoenterostomy, which we are confident would, because the benefit of a bile duct, the reason you have this long tube there is so that fecal content from the gut doesn't reflux up your liver, because that will give you cholangiocytes, which all of PSC patients know very, very well. So so you would need two years to get MHRA approval at the best circumstances, or say 5, and then another 2 years to do the first safety trial, at which point you would go to your efficacy trial for another 1 or 2 years. So I think it's between 5 and 10 years, to be honest, before you can get it the same way the first infliximab infusion started.
And as well, it's assuming that you can get the funding for all of this too.
Yes, but hopefully, I think if you support it quite well, there is so many, there is so much push, both from UK research and innovation, from companies. You know, it's something that I think if the research is good, you get a lot of people behind you.
Yeah,
but funding is crucial.
This has got so much potential. You can see, you know, it's not just bile ducts, is it, that this could benefit?
No, that's why it got a lot of traction, because that happened in bile duct, and the bile duct is, the beauty of the bile duct is that it's relatively single layer of cells, and there are vessels, of course, but these the animal or the human body provides the vessels because they start going into the scaffold the minute we put it in. But people are now thinking about it for more complex structures. For example, there's amazing work which is being done by Paolo De Coppi in UCL for pediatric bowel grafts along similar lines. You know, or we inject cholangiocytes in the liver. In the future, we will be able to inject other cell types. We will be able to do the same for hepatocytes. You know, it's I think it's one of these things that gets things in motion. And what is more important is that if we go to MHRA once and say we use the human cells as organoids, can we take them to trials? Then what PSC patients could help is everybody who has organoids in the future. Because the second person who comes on and says, I want to take bowel organoids in trials, they will say, how do you know it works? Well, the previous guys did it for PSC, you know, it worked. And so it'll be much easier for many other patients.
Yeah, so pioneering for PSC.
Well, I mean, if it works, right?
Of course it will. No, that's, yeah, I understand. You have to do the research and design the trials well and so on, and take it, not rush, I guess.
Absolutely.
So next question, is norUrso looking like it could be a future treatment for PSC based on findings that it may target inflammation in the bile ducts?
This is a very good question. We're really interested in it. I don't think we've got the definitive answer yet. I think one of the best persons to talk to about this is Michael Crowner, who's an expert on bile acids and is doing a lot of research. And hopefully we have a leading member of the Crowner lab, Claudia Fuchs, who's going to be visiting our lab. So this is work that we will be starting on the 15th of October. So
wow.
Soon. But of course, clinically, we are not answering this question. We're trying to look at what's happening in cells. There are trials going on, so hopefully we'll have data soon.
this actually leads beautifully onto the next question, because it is about what's happening in the cells. So do you have any theories or views on what causes the most damage to cholangiocytes? For example, is it being damaged by our bile acid or the immune system, or a combination of both, or something else? That's a good question.
I have to be honest, this is part of what we're doing. So we didn't touch upon the mechanism of the disease modeling, but we don't know. And it very much, and everybody has their own views. So, and definitely when you look at studies where they've done genome-wide association, which is a way of looking at factors in the genetic material that, or pointers from the genetic material that could lead us to what could be wrong, what changes in patients with PSC. Definitely, there seem to be changes which are associated with the immune system. So there's a very big school of thought that's working towards that. And our collaborators in Oslo are setting up, and in Birmingham are setting up to do a series of co-culture experiments whereby now we introduce immune cells around the organoids to see what's happening. There's another school of thought that says that bile is toxic in PSC. And we have seen that in our own experiments. I do believe that there are changes in bile in PSC. And that could be the cause of the disease, or it could be associated with the gut microbiota, because a lot of the metabolism of the bile acids get changed by germs in our bowel. And then they get reabsorbed. So the body has a way to preserve bile acids. So they go through bile in the gut, they digest the food, and then the body doesn't want to lose them. There is a mechanism to reclaim them when they reach the end of the road at the bowel. So it doesn't get wasted with bowel motions, but it gets reabsorbed at the very last minute. However, sometimes germs in the gut can change the bile acids as they go through. And as they modify them, they modify the composition of bile in PSC patients. Now, there's a lot of evidence, well, there's some evidence, I should say, that changes in the composition of bile do play a role. We are very big fans of that theory. We haven't proven it yet, but we are definitely working on it. Having said that, we saw that urso definitely doesn't make a huge change for PSC patients, at least when we look at them as a uniform group. On the one hand, we say that bile plays such a huge role. On the other hand, when we make bile more mild, more hydrophilic, we don't solve the problem. And it's a very big question why this happens. So one element is the immune cells. Another element is the microenvironment and bile. The third thing that we touched upon is the composition of the germs in the gut. This is getting a lot of traction, the microbiota. And this is why I get a lot of questions about patients in clinic. Does what I eat affect my PSC? I am not an expert on that, so I can't really answer the question. But I don't think that it is as straightforward as that because we get one food group and we include or exclude, we can cure PSC. And unfortunately, by the time we know that PSC is there, a lot of the changes are established already. So who knows if we had a crystal ball and we knew at the very beginning. And then there is a lot about the fibrosis, the scarring. So there's a lot of people who say, is it a problem between the interaction of the bile duct cell and the cells that lay down the collagen? So unfortunately, it's all a black box. And if you ask me, I think it's a little bit of everything. And there's a very good review in the Journal of Hepatology by Tom Carlson saying exactly that you get tiny, tiny contribution for each and every one of these components.
So if you replace the bile duct or a section of the bile duct, the same forces are going to be working on those cholangiocytes to maybe bring the PSC back or, attack them, I guess.
This is a brilliant question. And you know what? It's not a hypothesis anymore. You're spot on. That's why PSC recurs in a third transplant, right? So it's not just, it could be that the cells are problematic, but we know that when we put a new liver in, something is amiss because it comes back. And this is just the immune system Well, it could be, but we know that we hit our transplanted livers with a lot of immunosuppression, and still PSC comes back. I think we're missing a link there. I don't know, Teresa, what do you think?
Well, I think you explained very clearly the fact that, of course, it's multifactorial and there are multiple factors. And what we can just try to do is isolate each and every one of them to try to understand if there is one of these factors that has a higher, stronger role or not.
Yeah, ultimately, I think that even if we replace the cells, we will have to top it up with something, be it a bit of immune suppression if we find a component of the immune system that is responsible, or be it a change in the microbiota with probiotics, something that will not only undo the damage that is done, but prevent the damage from coming back.
Yeah, there's a lot. It's not called the black box for nothing, is it? It's a very tricky disease. Now, are you okay to carry on, because we've got quite a lot of questions?
I'm absolutely fine until 5.
Okay.
So, but it's up to Teresa if she's.
Yeah, of course.
And up to you, of course, because we're taking.
Oh, I'm very happy to as well. So let's carry on. So thank you. Okay, next question. How long do the new cells take to grow, and is this a determining factor in deciding if there is time to do this kind of treatment? We mentioned this a little bit earlier, didn't we, when we were talking about how long it took to fill up the little collagen tube?
Yes. So the cells in the lab. Well, I'll let Teresa talk about the cells in the lab.
Okay, just pitch in at any time. And as we discussed before, what we see is that we have never seen much variability between different patients. Therefore, we can quite confidently say that from the time we get a sample, we will have to wait a few weeks before we have the critical mass to then be able to perform an injection or create a bioengineered bile duct. And yeah, of course, that depends on the type of technique that we need to use. Of course, if you want if the generation of bioengineered bile duct is the final scope, you will need a certain amount of cells, and it will be different for an injection.
But I think it's a very insightful question, because there's data showing that the cells in PSC and in a lot of bile duct disorders they have a property which is called senescence. So when the cell senses that it is sick, it focuses on healing. So, if you've got a cold and a cough, you won't go for a run. So it stops proliferating and it becomes slower. So what we have seen is that in our animal models, damaged cells tend to proliferate less fast. But what we also see, which we are trying to understand, is that when we take PSC cells and we grow them in the lab, they forget to be slow. They sort of catch up. So clearly there is something in the body that slows them down. And the expansion of the cells, so it's a very good question, because initially I expected that we wouldn't be able to grow the cell. So expansion from a patient's cells to repopulate the duct or to put them in the body is not a problem. But when they go back in the body, I suspect that they will be slower. But again, watch this space. There are so many questions, and I think this is another one for our note.
Yeah. They are really good questions. There's another great one here. Do you see this as being a possibility for treating recurrent PSC, which is often identified at an earlier stage?
Yeah, I think that that would be a very good option, and it would be a very good option for. I suppose when you say recurrent PSC, you mean after transplantation, and that would be a very good option, because it also opens up the option to use any cell we like. So if we wanted to do patient cells at first, if it's a transplanted liver, there's absolutely no reason to do that. So we can use healthy cells or cells that we have modified even in some way, which we keep as a backup, which we keep in a biobank and sort of can then for the treatment of recurrent PSC, because you would already be immunosuppressed. You wouldn't stop your immunosuppression. You have a new liver. You have to protect your gut anyway because of the rest of the liver. So absolutely, and I think it's absolutely fair to see that we are more and more vigilant about that. And after transplantation, you get so many scans, so high surveillance.
It's just thinking about that, the rejection issue. It makes perfect sense, doesn't it, hypothetically, to do a transplant for PSC and then grow beautiful bile duct, common bile duct, to connect the new liver to the body. Yes, surely. And without having to do this sort of the joining up that they do already for PSC transplants is not very it's not as nice and straightforward, is it, as usual livers with other liver diseases?
Yeah, so that historically has been an issue because we think that there is higher chances of stricturing happening. Now, however, having said that, because I need to be 100% honest, I'm in two minds about that. On the one hand, I'm thinking that what we do is we take the healthy liver, the new liver that comes in, and we cut out the bile duct. Why? Because we are so worried that we're going to get a stricture at the bile duct. But you know what? And this is where the disease is multifactorial. This is a healthy bile duct. So I think we absolutely could use an artificial bile duct to avoid doing the biliary reconstruction and bringing the bowel close. But you know what I think we should be doing? I think that since it is artificial, we should modify our scaffold a bit to be less prone to fibrosis. And this is part of the research that we are doing, because if a healthy bile duct still gets it, still gets that stricture after it's going in, if you put a bioengineered bile duct, an artificial bile duct with healthy cells, most likely it would still be high risk for getting the stricture. What we need to do is find out why it happens in that healthy duct and figure out why and how we can avoid it. But again, it is a brilliant question because it brings the idea of, should we now be sampling bile ducts? Well, we don't do it anymore anyway, but it would be great to see if we had some samples of patients who have had a transplant and got a stricture which hasn't got to do with the operation in that bile duct. So if we could go back to this historical cohort of patients and see what went wrong in their tissue, how come they got a stricture when their bile duct was essentially new. But of course, having said that, to do that, you need a stamp of the native bile duct, right? So
there would still be some of the original one in there.
Yeah.
You've a lot of questions to answer, like for research questions to answer as well as here. Okay, this is a question about, would a genetic test for PSC help with this treatment as it requires an early diagnosis?
Yes, I think if we have biomarkers, and this is why we can't do this on our own. PSC research is not isolated. It's a pleasure to work on it as a scientist because you understand that you have, it's not for children who don't like to share their toys in the playground. You have to work together. So yes, absolutely, we need a marker that tells us not only early on that there is disease, but also, we discuss about all these horrible complications, but the truth of the matter is that a lot of the patients will carry on to live a perfectly healthy life. And will go through their screening and will go through the normal steps, and they will never have a problem. Or they might get a gallbladder polyp and need to have their gallbladder out at some point, but they will live an absolutely normal life, right? But we have no way of knowing if you are one of the few less lucky ones where something could potentially go wrong. Now, giving you cells, it's one of the latest treatments, right? And it's not easy. And there's always a risk. Anything that you haven't tested, there's a risk with. But if we knew that your disease was going to progress and be aggressive, that's a very good argument to give you a new treatment. On the other hand, I can understand if people would be reluctant if they came to us and said, have I got PSC? Yeah, it's very early. Shall we give you cells? What are the chances that I'm going to need them? I don't know, 20%. So then they say, so you 4 in 5 chances I will get a treatment that I don't need, that might cause complications, whilst I could go on and have a healthy life without any problems anyway. So you see, this is why it's not only important to know when the disease comes on, but also how aggressive the disease will be, how it will project. And that's why I'm super supportive of all the research in biomarkers, in early diagnosis, and what we call precision medicine. I think we should really support.
No, I agree. And and if we had decent biomarkers, we'd be able to make a better risk-benefit judgment about whether or not to take some cells. And I think that different patients will have a different appetite for that, and some will be, I want knocking on your door to have it, and others would say, well, let's just wait. But it's a good reminder to us all that not everybody gets these advanced, not everybody gets fibrosis and cirrhosis and complications, but it's for the ones that do that we're trying to get trying to do all this.
Yeah, absolutely, because you never know when you might, there's always tomorrow, right? And
Yeah.
so you know that everything was fine up to today. That's why we are so keen to keep on screening. And this is
Good.
why when we see patients in clinic who are especially very young, when you're young, you live a very busy life, right? You have a lot of things to do, and it's sometimes very difficult. You are quite mobile. You might move from one place to the other for studies or for work. But it's crucial, for example, that you keep up your ultrasound screen every few months or so. That's
Yeah.
it, because you never know.
Yeah, I think that's one of the hardest things about PSC, regardless of what stage you're at. It's the uncertainty, and it affects all of us.
I agree, and I really hope this is one of the things that I really hope to be able to make a difference on. Not necessarily us, but generally us.
Yeah. Yeah. No, I agree. Okay, another question. So, is the immune system letting in germs that should be there? Changes the composition of the bile, so the body doesn't recognize the things it needs to keep, and that becomes a problem. The ducts are dealing with bile that they shouldn't. I don't think I've read that shall I read it again? So, is the immune system letting in germs that should be there, changing the composition of the bile so the body doesn't recognize the things it needs to keep, and then that becomes the problem the ducts are dealing with? Are the ducts dealing with bile that they shouldn't be dealing with? I think this question was probably written when we were talking about all the different potential causes of PSC. Yes.
I don't know, Teresa, what do you think?
Well, I think it boils down to what we were saying before, that probably it's more of a complex mechanism, whether the immune system sort of is less effective in keeping a barrier for some germs.
Yes, exactly. So I have to admit, I'm not an immunologist, so I may be saying something wrong. So I think that it's not just the immune system that controls what type of germs are in our bowel. It's again quite multifactorial. But here I'm not an expert, so I can't go through all the factors. And I'm sure that it could be that the immune system has some role to play. But I think I'm not the right man to answer this question because it's way out of my head. But all in all, I think ultimately, if we get a population of germs that treats bile acids differently and changes them, that will definitely, as Teresa said, could add to the problem if they are what we call bad bile acids or more toxic bile acids. That certainly is going to contribute to the damage. I think because there are so many things that contribute, it boils down to what initiates it, Because, how shall I say, you put diesel in a petrol car, right? There are so many noises. You can take it to the mechanic and say, but now the gasket is not in a good condition and there are noises and my exhaust pipe is giving you. But the problem is that you put diesel in a petrol car, you know, that's where you need to start. You can fix everything else in your engine, but as long as you keep fitting it diesel, you'll just be replacing parts that break every time. So I'm sure it is absolutely a contributing factor what happens with the microbiota and bile acid and so on and so forth. I think that the most difficult thing is finding the initiating factor in all these aspects. I'm not sure it entirely answers the question. I'm sorry.
I think it does, and I apologise to Christopher for the way I read it out. I didn't read it out very well, but you answered it well, so thank you. There's some really lovely comments now about how helpful this meeting has been and how informative. Let's see what other questions we've got.
Thank you.
Would it be possible to design an experiment to take bile from someone from PSC one day and apply it to the bile duct in the lab whilst monitoring their diet to see how reabsorbed bile affects cholangiocytes based on diet? And this would also take the immune system out of the picture for the purpose of this experiment. That's a really good one.
We need to hire someone here.
Almost you want, when people are on drug trials as well, we should be saying like they need to get a biopsy and they need to give it to you to test to see what the before and after is like for the bile duct cells.
Exactly. So as Teresa was saying, it's a very cool idea, and it's one of the things that we are doing. We have collected some PSC files, and we're trying to do some experiments. And what we found, this is the basis of one of my most recent UKRI grant, is we find that cholangiocytes from PSC patients show some changes, some susceptibility, which is only coming forward when you treat them with PSC bile. So there's a million things that go wrong, but we have some early evidence that PSC bile might also play a role. In other words, if you change the bile composition, you might be able to make things better. But again, UrSO, I have to stress that UrSO changes bile composition. We haven't seen an effect. So we need to figure out exactly how. So hopefully Teresa will answer that question for me. And then
we'll wait for your paper, Teresa.
Okay, no pressure.
No. Okay, next. Oh, someone said this has been one of the best live sessions they've ever joined. So they really show their appreciation.
It's been massively helpful for us, right? As I said, we took a lot of notes.
We really appreciate it. Let's see what other questions there are. Oh, Christopher said, sorry, writing that one as they were talking. So you know the question I read out wrong. Yeah. He was writing it as you were talking. This is a good question.
No problem at all, of course. It's a very good question.
Okay, we've got one last one now. Regarding injecting the bioengineered cholangiocytes, when the cholangiocytes are damaged due to disease, scar tissue builds up in the bile duct wall. Is there a point where too much scar tissue builds up, preventing the bioengineered cholangiocytes from sticking?
Yes, so that's a very good question. That's why I think, so we don't know for sure. There's a very high chance that this will happen, right? And of course, if you have a stricture, there's a chance that cells won't be able to get past it, to get above. So, the rest of the bile duct will be inaccessible. There's a chance that because collagen, after a while, it's the same thing as with your skin, right? At first, you get the scar is raw, and you see that the tissue is still healing. Once it's healed, it closes, and then that's it. You've got a scar there, and that scar is very smooth. So I agree with you. We haven't tested it, but my gut feeling is the same as what you are suggesting, that after a while, it will stop being sticky for the cells to stick, and that might be a problem. And we have some preliminary data suggesting that, because we know that an old scar has different types of collagen compared to a new scar. And there are people who do a lot of work on fibrosis and fibrogenesis that are great teachers, and we hope to get their views on that. So, as I said, That's a very good one, and it's something worth testing in the lab. In addition, we think that once the stricture has been established, even if your cells were to stick, it wouldn't go away, you know. The tight area, the fact that the lumen from this big is now that big because it was strangulated by scar tissue around it, will not go away by virtue of stopping the attack of bile. Because, okay, it won't get worse, but it won't get better.
Yeah.
So I think we need something there to help us change the scar tissue as well, if possible.
Do you think you'd be able to combine it with, like, you know, when you go and put stents in or do balloon dilatation, could you combine it with that? So if there was a particularly scarred section that you could get to, would you prefer to snip that section out and replace it if it was accessible, or make it wider and then put the cells in?
That's a very good question. It may be worth discussing that with Gareth, Teresa, because I don't know. You know, that's a very good question, because
I'm going to be coming to work with you on Monday, just so you know.
We're hiding.
I'm bringing everyone with me from this.
Yeah, that's a good point. And because it will be very interesting to see what is the process of reformation of a stricture after balloon dilatation. Is it the fact that we broke it, but still there is attack of bile, and through the same process, and the stricture comes back? So yeah, it's really cool now that you mentioned it. Should we be coating the duct with cells after we dilate the stricture? Could it give us a benefit? I don't know. Why don't we do it and see what happens in the lab? So that's a big question.
So thank you, and I think I've asked all the questions now, and apologies to anybody if I've missed any or not asked them right. But I think that we'll all agree this is a really incredible time for PSC research. We've got clinical trials testing drugs going on, and we've got bile ducts being built. It's amazing. So I really hope your research continues to thrive, and we're always happy to try and contribute and, you know, give our opinions and so on, if that would help your research. So thank you very much, both of you, for talking to us today. We really, really appreciate it. And there's such excellent comments and signs of appreciation coming on the comments now. And so thank you everyone else who watched and asked questions. There were really good ones today. Enjoy the rest of your weekend. Teresa, I hope the weather's better tomorrow and that you have a really good...
difficult.
I hope your run goes really well, and to everyone else that's running the marathon, I wish you all the best for that. And with that, we will leave you all. So thank you very much, everybody.
Thank you very much. It was super helpful.
So much.
Thank you.
It's been brilliant. Thank you.